there is an urgent need of antivirals and vaccines to protect global population against COVID-19, which has already reached to 40 lakh people and killed 2.8 lakh of them. Different strategies are being worked out. Recombinant BCG is one of them. Proponents of recombinant BCG say that if they are genetically engineering, they are genetically modified version of BCG can help protect people from COVID-19. To discuss this crucial topic, we have today with us Dr. Gobardhan Das. He is molecular medicine. He's from molecular medicine department at JNU Delhi. We have Dr. Om Shrivastav. He is infectious disease expert from Mumbai. And we have Dr. Abel Lawrence from SGPGI Lucknow. Welcome you all to the show.
Thank you.
Dr. Das, so we heard that you were among the earlier proponents of recombinant BCG in India. And I heard that you had submitted a proposal to the apex approval bodies, but apparently it was turned down. And then we heard that some of the Indian institutions are working on the same projects, like Serum Institute and Hampton Institute. So both are going to start trial now. And also we heard that now your project has got approval from an international body with $7 lakh grants. So can you please explain how do you propose to develop this recombinant vaccine for COVID-19?
Well, when this COVID-19 is spreading throughout the world, just out of China, so myself and my colleague, Dr. Anand Ranganathan, so basically we are discussing and looking at the data coming from the several countries throughout the globe. What we found, the only one common thing, whether they have the BCG immunization or not. So basically, over a period of time, within next 1 week, we compared data with 178 countries, and it turned out to be it as a perfect inverse correlation whether the country has the BCG immunization and versus the COVID-19 incidence as well as mortality. So then immediately I started writing to our government. Also, another thing I forgot to mention, last 2 years, there are intense research on BCG immunization and the innate memory. So from those paper and the current data, the spreading and the incidence as well as the mortality, what we correlated, there must be something with the BCG vaccination. And that's because, as you know, the BCG vaccination, BCG is a vaccine intended for tuberculosis. So, but as you know, this is BCG can protect children from disseminated TB and meningitis, but the immune response over a period of time goes down. So, but in countries like India, Brazil, so they have hygiene index is much less, and probably we are being exposed to various organisms, but somehow we are keeping that memory alive for a long time. So then I thought, why not to BCG reimmunization? And if with the BCG reimmunization, in the meantime, we also work a lot of work done with the MIP, something called Mycobacterium indicus pranae. So which is a little better than BCG. Then I started talking about why not immunize with MIP, which is also known as MW. So I wrote to minister also. I wrote to ICMR DG also, and then also in the meantime, I thought we need little more specific vaccine. So how do we make this specific vaccine? As you know, BCG is a bacteria, whereas COVID-19 is a virus. So basically, the immune response that is protecting has to be the non-specific. So how do we get a specific vaccine? How do we make it specific to COVID-19? Then the idea came that what are the immune responses we need for the protection? We need, of course, doctors. They all this is we need type 1 interferon, innate immune activation, cytotoxic T cells, and the antibodies. So therefore, what I thought, can we get better cytotoxic T cell response? Because the BCG does, but it's little weak. the A recombinant BCG strain is available. What is in where actually LLO, the protein which is coming from the Listeria monocytogen, is recombinant in BCG that gives better cytotoxic T cell response. Then I thought, on this BCG, can we put some COVID-19 protein so as to get the better cytotoxic T cell response as well as the antibody response. So basically, having the COVID-19 protein on this strain, we are going to get it the basal immune response, such as the type 1 interferon, innate immune activation, then antibody response, and the cytotoxic T cells all together. So that was the kind of proposal I made it, and then I thought this it should go in a very high speed. That's what I wrote it to our science advisor, and he asked me to put it in one of the granting agency. I put it in and turned it down. I thought maybe it's not a worth to continue. So somehow then I have to go fast. Then I send it to some international organization, and I gave it to them. And I was very surprised. In less than 24 hours, they say, "Don't worry, you can take $700,000, and for next 3 months, and then we'll take it in the high speed." Okay, great.
Dr. Srivastava, as we know that BCG vaccine has not been very effective, in protecting against tuberculosis, because India has got largest TB population. So we can see its effectiveness. So first of all, why it is not so effective against TB? And in that case, whether it would be able to provide immunity against COVID-19?
Okay, so two separate questions you're asking. Why is the vaccine not as effective against tuberculosis as we think it should have been? Well, tuberculosis in itself, when we say Mycobacteria tuberculosis, there are number of subparts that can cause infection. There are, without going into too much technical details, there are what we call the slow growers, the rapid growers, the Mycobacteria other than tuberculosis, which is the not bacillus. So Mycobacteria is not one name; it is a number of subspecies which have mechanisms to try and protect themselves from a number of things, not just medication, but also the vaccine that you just mentioned. Over the years, Mycobacteria have done this protection mechanism to be so robust that unless you've got a vaccine that will keep covering all these parts of Mycobacteria, it's not going to be effective. You often say that Mycobacteria tuberculosis is...
Yeah, I would like to add some things regarding the BCG vaccination. There are interesting data on the immunomodulatory effects of BCG. For example, I had come across a study from Nijmajan, Rob et al. Group, which looked at the effect of BCG on this yellow fever, attenuated virus strain of yellow fever. And what they found was that when you give BCG vaccine along with the yellow fever vaccine, it leads to enhanced immune response against yellow fever. What they noticed was that this attenuated vaccine strain, which causes viremia in the blood, the level of viremia was less in people who received BCG. The interesting thing was that this did not lead to any changes or differences in adaptive immune response, T cell responses, or antibody response or anything, but the viremia level was lower. And they found that this lowered viremia was a consequence of altered innate response, which is what we are calling a strained memory. That is, some people call innate memory. I have a problem because when you teach people about innate immune system and adaptive immune system, you talk of innate as something not having memory, but this trained memory is the term they have used. In trained memory, what they found is that BCG vaccination leads to increased IL-1 signaling, and which leads to increased IL-6 signaling, and epigenetic changes in monocytes which mediate all these things. The issue here is that they did not find any changes in type 1 or type 3 interferon response after BCG vaccination, which is very surprising. You would expect something like this to have that. And when we look at the immune response to coronavirus, the striking thing is that the coronavirus evades the interferon response. The type 1 and type 2 interferons responses are also low, and there is decreased MHC class 2 expression, which means that the type 2 interferon response may also be low. And there is an exaggerated IL-1 and IL-6 response. So much so that tocilizumab is being used as a treatment for complicated coronavirus disease. It has been approved in China. And there is a recent study which found that anakinra is also useful in decreasing complications of coronavirus disease. So therefore, what we have noticed is that more than the virus itself, it is this exaggerated IL-1 and IL-6 response, which is a problem in coronavirus disease. And we have this BCG vaccine, which augments the IL-1 and IL-6. It may actually contribute to pathology. This is a concern which I have. And coming to the protection of BCG, subsequently, when the same country-wide data was analyzed after adjusting for the timing of entry of the virus into countries, thus its effect disappeared. And if the effect is true, you should expect people from subcontinent who have been vaccinated against BCG to have some protection. On the other hand, if you look at the medical personnel in UK, those from the Indian subcontinent, Africa, who have been vaccinated actually had poor outcomes. The death rate of medical professional from the subcontinent who vaccinated was worse than the whites who are not vaccinated. They don't vaccinate people in UK. So this, so if the vaccine is actually protective, then vaccinated people should be protected in places where both live together. Countries have other factors, the connectivity, airlines, and in those stochastic factors can play a role. And there has been a subsequent analysis from Japan, where if you are taking this thing seriously, then why did Wuhan in Hubei province in China, where they vaccinate people with BCG, why did they have such a large outbreak? How did Korea have outbreak before they managed to control it? So the differences in between countries can be best explained by politics and social factors and timing and connectedness rather than any BCG vaccination. Countries which took coronavirus seriously, like India, Korea, and most of Asian countries, have done better than countries like the Netherlands, UK, Sweden, and US, which did not take it seriously. What we find is a political divide than a divide along the BCG. And the BCG effects may just be a matter of chance.
Dr. Das, yes, please.
Yeah, so let me answer this question. He raised a very valid question. So number one, so when we are talking about the vaccination, first question, when we are talking about vaccination, in no way the patient who are in ventilator, having severe disease, the vaccination is not going to work. And not only work, there are two reasons. As you know, all COVID patients, they have something called lymphopenia. In the periphery, you won't see the very few number of lymphocytes as well as this phagocytes, macrophage, monocytes. So all of them actually mostly translocate to the site of infection and where the huge inflammation. So if you certainly if you immunize with BCG or anything which mount immune response may not work at all. So that is what you need a team with not only the doctors but scientists and many other fields. So that is what one of the things one has to think. Similarly, antibody response. People keep on talking about well, we are going to transfuse antibodies. Those who are in the serious conditions, they're never going to work with the antibody transfusion or the replacement. That's because already patient they themselves have the antibodies. No, so similar thing. Coming to the his second question, the countries like China, Wuhan, China is also vaccinated with the BCG. I'm sure you know that the China's vaccination program is severely impacted during this great famine time, and that time not only the vaccination but also food habit and the malnutrition, many other things that can hugely affect. So, and second thing I would like to raise the question: we are talking about a pocket of a whole country. So basically, we really cannot compare data of a small pocket with with respect to the big countries. And fourth thing I would like to talk about, in our data, this paper is now in under revision. Our data, what we saw is not all the BCG strains in the work equally good. So, for example, what we use like in a mixed strain, as well as some countries use Japan strain, which is the best, whereas some other BCG Denmark, which is the worst one. So basically, that also one has to consider it.
Dr. Das, he also mentioned about IL-16 and IL-1, right, Dr. Lawrence?
IL-1
yes.
and IL-6. They in fact link the differences to IL-1 polymorphisms and histone acetylation in the PI3 kinase epidermal growth factor subgroup. IL-1.
Yeah, so sorry, I forgot to tell that you mentioned that. See, these are the IL-1, IL-6. These are the indeed they're the inflammatory cytokine. So inflammatory cytokine, and then the type 1 interferons, the type 2 interferons. Of course, they are the inflammatory cytokines. Absolutely, I agree with you. And these are the inflammatory cytokines. Inflammatory response actually exhibits the disease, and that is what most of the patient is dying. That's true. But before, when the virus actually is coming in your system, before it adapts in your body, or make a shedding in your body, or hiding in your body, so these are the inflammatory response actually eliminates the virus. So once again, I would like to mention here, once your disease is onset, already inflammation is already started, and there is lymphopenia, I think there's no use of vaccine. But same inflammatory response, the moment the virus comes in your body, the immune response, makes a little inflammation, and that can eliminate the virus. However, if the virus goes and your adaptive immune response takes time, and it will take 8 to 9 days by the time the patient was in the severe conditions, that you really cannot rescue with you cannot do the vaccination with the severe patient.
Dr. Srivastava, do you think this recombinant BCG is going to work? Because I asked you earlier, your audio was not audible, that when BCG vaccine is not very effective in protecting people from TB, then how can it provide protection in COVID-19?
So I'd like to just go back quickly on the comment that Dr. Das was just making, that lymphopenia and the total amount of the cytokine response have a mismatch. So I think that clinically, I'd have to say, when you measure the levels of interleukin-6 in patients who are very unwell, who've got lymphopenia, you'll find that interleukin-6 levels in the range of 100, 120, 150, sometimes as high as 3000s have been seen to occur in patients who've got a white cell count of 3 or 2 or even less than that. So I think there is a mismatch in the total amount of lymphopenia or cytopenia and the total amount of interleukin-6 levels. So there is clinically, at least, there doesn't appear to be a correlation where you can say that because your patient's white cell count is sitting in the lymphopenic range or cytopenic range, the interleukin-6 response is going to be subdued as well. Clinically, we found that there is a huge mismatch in this kind of situation. Coming to the question that you've just asked me, will the recombinant BCG vaccine work? Well, there really isn't a role of the vaccine when a patient is already into a stage 2, stage 2B, stage 3, which is the time where the damage has already set in, or it is beginning to set in. The damage involving the lungs or the blood vessels or the coagulation factors, all of that has already come to a stage where it is in a state of intense inflammation, and that at that time to give a vaccine to mount an antibody response, that is a complete, well, that strategy in itself is flawed. So I don't think there is going to be a role for giving the vaccine at a time when a patient is unwell. What will work is probably to position a patient who is likely to be at risk, or for a community to take the vaccine if you think that the second wave or the third wave or the fourth wave of the coronavirus is going to be coming into the community in the next maybe 2 weeks time, 4 weeks time, 6 weeks time. That is the time to start giving the vaccine in the recombinant state, in the hope that it is actually going to be giving, as was already being pointed out, that you are going to need to get that part of your patient's innate immunity to develop those antibodies that will be part of a memory that will prevent the next wave of the virus from causing catastrophic complications in a clinical setting. So that's probably the situation where it is likely to work.
Dr. Das, which population is targeted at by your vaccine?
Well, initially we proposed those who are in the high risk group. For example, the medical professionals, those people, those who are taking care of COVID patients at home, and those who are having preset other kind of disease, and the elderly population. So that is what we thought. I mean, I agree with Dr. Srivastava that already those patients who have onset disease, vaccine is not going to work. So that is not only for vaccine but also for the antibodies. I think, my opinion. And also, I would like to answer Dr. Srivastava about IL-6. You know, IL-6 not only is produced by your phagocytes or macrophages, but also various epithelial cells. And also the amount of IL-6 or the other cytokines produced in the lung that may have access to the circulation. So you may see this as, hyper, IL-6 or interleukin in the periphery. However, that may not be the local here in produced in the local in the periphery. That may be produced in the lung itself. So by being a doctor, probably you know much better than me. I'm not a I'm a scientist. I'm not a doctor.
Dr. Das, I would also like to ask you at what stage your project is at present. Have you already genetically modified the vaccine, or I mean, how much time it is going to take?
Well, for me, actually, right now here today, what we did is we did all this the construct, and we are at the stage of cloning. So basically, we put all these the COVID-19 proteins in the vector. So basically, in next week or so, we'll be able to transfect, and then colony selection, and then goes in the animal. So basically, I don't know, maybe another couple of weeks we will be ready. However, in the meantime, we needed some grants, so that is what we are looking for it. Right now, we got it, and then we are continuing that.
Which part of the coronavirus antigen you are looking at?
Well, that is initially we started with a spike protein, and the spike, and then AM, and then nuclear proteins, and there are five of the proteins we have taken, and then we put it in individually, put it in vectors, and then put it in BCG or the modified BCG, and then from the animal study we'll find out the epitope, and then we'll take out all the epitopes and put a new antigen, so that all the HLA, all the various HLAs are covered. So that is what, and then when we get a new synthesized vaccine or antigen, that will put it in BCG or modified BCG. So basically, down the road, what we are thinking, we'll get it maybe in a 6 months or so.
Dr. Lawrence, you might have heard about other vaccines like the one proposed by Oxford, which has already gone under human trial. And then we have the BCG vaccine trial and this recombinant BCG. Which one do you think is going to be most effective or going to come first?
When we look at a pandemic, the most important thing is not how effective a vaccine is. How fast can you get an effective vaccine? So it is a more of a race. Even if you have a very effective vaccine which provides robust immunity, if it comes too late, is of no use. So basically, vaccine development requires quite a lot of time. Most of the time, successful vaccines are projects that run 4 to 5 years. And if you have adequate funding and everything is done properly, everybody says it's minimum of 18 months for regulatory, and then manufacturing vaccine quantities is also a challenge. And in that aspect, the vaccines which were under development for the last 10 years definitely have an advance and are in advanced stages, stand a better chance of success than the ones which are being developed now. So in that way, as of now, from the information we have, the Oxford vaccine, that is recombinant adenoviral vector vaccine, where they have put a, the spike protein inside the chimpanzee adenovirus, that is already entered trial and can be easily manufactured in existing facilities. And if everything is going according to plan, it may be available by October. That is, if everything goes to plan. We may not happen. There may be some unexpected problems. And similarly, Pasteur Institute has developed this measles, that is measles vaccine strain based, recombinant vaccine against coronavirus, which also has a long history. They have been developing vaccine against SARS and MERS for many years, and therefore it's a matter of swapping. Both these vaccines had phase 1 trials with MERS and SARS antigen, so we know something about it, so they stand a better chance. However, nothing can be sure. And regarding this BCG, and I have to point out another fact. In India, almost everybody is vaccinated against BCG. So therefore, when you give BCG to people who are already immune to tuberculosis or have vaccine, then there can be serious reactions. The adverse events, ulceration, complications actually go up. So this may make it acceptability of, even assuming that BCG vaccine against coronavirus is effective, this may be a major problem.
Dr. Srivastava, your concluding remarks, please.
Yeah, I think that while we are all talking about the potential of a number of vaccines that are now coming to this phase, the phase 2 or then going on to the phase 3, that's going to require very careful interpretation of the people who will benefit from the vaccine. So I think that while we are all very keen to find out whether a vaccine is going to be available, I mean, Abbott is talking about maybe as early as the end of June or beginning of July. How much these vaccines are going to be effective for the largest population that is going to be needing them, we will have to wait and see. Already, if you go to some of the sites that monitor and track these things, there are already about 32 or 33 variations of the genetic fingerprinting of the virus since the time we've been tracking it from January of this year. So in a period of 5 months, there have been about 30 odd variations, 30 plus variations. I'd like to see a vaccine that covers at least, if not all of them, at least the most part of these variations, because there's no point hurrying into a vaccine that is going to be offering protection only to 20% of the people. You'd much rather wait and see whether a vaccine will cover these strains and offer protection to about 60-70% of the people. So I would prefer to wait for a few months than rush into a vaccine only to find out that it is not effective. I think these are complex issues. They should be dealt with in a lot of complex procedures so that you are able to do the best to the largest population.
Dr. Das, quick concluding remarks, please. Very quick.
Yeah. So I really agree with Dr. Srivastava that another important point I forgot to mention, as you know, this is a flu virus. They doesn't have memory. Every year you have to revaccinate. So basically, those short vaccine, those who are making in the adenovirus probably may not have good memory. In that case, for